In the advanced molecular biology course, we will study with the cell cycle control system and how it regulates itself. The cell cycle is a very complicated process, and in this lecture, I will try to highlight some of the points for the cell control system. Now, the cell control system, the sequential events of a cell cycle are needed to be governed by a system where, for example, in this cartoon, we have discussed that the cell growth occurs first initially, and then the chromosome segregation occurs, and then the cell division occurs, and another daughter cell is produced. Cell cycle control system, there are two irreversible points in the cell cycle which have to be kept in mind. One is the replication of genetic material and the separation of the system chromatid. Checkpoints in this process are that the cell cycle is controlled by stop and go chemical signals at critical points. Signals indicate if key cellular processes have been completed correctly. Now, there are two phases of a cell control system. One is the S phase, and the second one is the M phase. S phase starts from the DNA synthesis, and the M phase, it is abbreviation of the mitosis phase, where there are two components, mitosis and the cytokinesis. In the cytokinesis, the cell, it breaks down into the two daughter cells. Now, cell cycle control system again operates like a timer that triggers the essential process of the cycle. That is the DNA replication, mitosis, and the cytokinesis. The switches which control these cycle, they are the binary on-off switches. Binary on-off switches are those switches which are initially just like on our light switch or in the car switches, and they launch event in a complete irreversible fashion. The system is highly adaptable and can be modified to suit specific cell types. Three major regulatory transitions during the cell control systems are: number one is the start. That is the start transition where the cell commits a cell cycle entry, and then G to M transition where the control system triggers the early mitotic events. And then there is a metaphase to anaphase transition where the control system stimulates the sister chromatid separation. Now, again, bear in mind that in most cells, the gap phases separate the major events of the S phase and M phase. Gap phases which are indicated like G1 is the gap between M phase and S phase, where the cell commits to cell cycle entry and chromosome duplication. G2 is the gap between S phase and the M phase, where chromatin is checked for replication. In this cartoon, these whole steps, they have been indicated at different points. For example, the cell system, it asks that the metaphase to anaphase transition, when there are all chromosomes are attached to the spindle, then there comes the G1, a gap, and again, then there is the S phase, and before S phase, there is enter cell cycle and proceed to S phase, then it goes to G phase, and it is all DNA replicated, and if the answer is yes, then the cell keeps on producing and keeps on its cycle of replication. Now the cyclin-dependent kinases, that the CDKS, that when cyclin forms a complex with CDK, the protein kinase is activated to trigger specific cell cycle. And without cyclin, the CDK is inactive. These are the major component of the cell cycle control system and originally named because they undergo a cycle of synthesis and degradation in each cell cycle. Cyclic changes in CDK's activity are controlled by an array of enzymes and other proteins. For example, in this cartoon, the cyclin CDK complexes, it has been shown that how G1 to G2 and G1, again these complexes, they vary and control the cell cycle. The major cyclins and the CDKs of vertebrates and the budding yeast has been illustrated. There are 3 D cyclins in mammals and like cyclin D1, D2 and D3, and the original name of CDK1 was CDKC2 in both vertebrate and fission yeast. Now the 4 classes of cyclins, these are G1 cyclin help to promote passage through start or the restriction point of G1. G1 over S cyclin, it activates CDKs in the late G1 and commits the cell to DNA replication. S cyclin binds CDKs during S phase and helps stimulate the chromosome duplication. And M cyclin, it promotes the event of the mitosis. So as we see, the cell cycle is a highly regulated and controlled process.